A steroid hormone passes through the plasma membrane and binds to and activates a specific receptor protein. A chaperone protein changes the shape of the receptor complex. The hormone receptor complex enters the nucleus and binds to a specific gene on the DNA. The round protein acts as a transcription factor and transcribes the gene into mRNA, which is translated into a specific protein.
Phosphorylation cascade, a signal molecule activates the receptor molecule, which in turn activates a relay molecule. This activated relay molecule activates inactive kinase 1. Active kinase 1 then phosphorylates inactive kinase 2. Then active kinase 2 stimulates the phosphorylation of inactive kinase 3. Finally, active protein kinase 3 phosphorylates an end protein that brings about the cell's response to the signal.
G-protein coupled receptors are a group of 7 transmembrane proteins. When the signal molecules bind to the outside of the cell, it activates the G-protein, changing the energy from GTP to GTP. The now active G-protein binds to the enzyme and activates it, thus causing a cellular response. Then, when the G-protein disconnects from the enzyme, it releases a phosphate before returning to its inactive form.
Receptor tyrosine kinase are cell surface receptors and function as receptors for a wide array of growth factors. There are first 2 receptor tyrosine kinases, and the signaling molecules bind to the signal binding site of the receptor. The first 2 receptors come together and form a dimer. Phosphate from the ATP is added to each tyrosine of the dimer, and the ATP changes to ADP. Relay proteins attach to the phosphorus and activate, causing a cellular response.